All right, so we are moving on to chapter 5. Chapter 5 deals with viruses and prions. You probably have never heard of a prion, so that'll be an interesting conversation. But viruses, I'm sure you're pretty familiar with. So when we talk about viruses, just to kind of put this out there, viruses are able to infect just about every type of cell. A lot of times we mainly think about viruses and how they affect us, right? So in humans, viruses cause everything from like the common cold to some of the more recent emerging diseases such as Zika and Ebola and COVID. Um, but they actually are capable of appearing in all ecosystems and can infect every type of life. So they infect bacteria as well. So that may be kind of interesting to you to think about a virus actually can infect a bacterial cell. They can infect algae, they can infect fungi, they can infect plants. You may be familiar with viral infections of plants. You may have heard of the tobacco mosaic virus. And the tobacco mosaic viruses causes a mosaic pattern on the leaves of tobacco and also tomato plants. And this was and still can be a very harmful virus for plants. And so it's kind of one of the first that we learned about. And even virus can be found in many of the protozoans. So some of the parasitic protozoans, like such as Giardia and Leishmania, these can have viruses inside of them. So a lot of times we don't think about that. So it's not just in humans that we find viruses, but viruses are capable of infecting just about every type of cell. So when we talk about a virus, right? So it's a very small agent. Um, so viruses are not cellular. So even though they're small as a cell, or and even smaller than a cell, they're very unlike prokaryotes and eukaryotic organisms because they are not comprised of a cell at all. They do not have cell walls, they don't have membranes, they don't have any organelles at all. So, because of that, and the fact that they require a host cell to reproduce because they cannot produce on their own, and a lot of times they need the machinery, like all the organelles that are inside of their host cell, to help build parts of the viral body. So they need to take over a host cell and use the host's organelles and amino acids and components that are found in a host cell in order to be able to make more viruses. So many scientists consider viruses to be kind of non-living because they cannot produce the components of their own cell, or excuse me, their own bodies. Um, so we can say they're non-living for those reasons. They need a host cell to reproduce. They're not comprised of cells. Now, on the other side of the coin, though, all viruses do evolve. So they are capable of evolving or changing over time. And we've seen this with the COVID virus and how many different variants of the COVID virus there have been. Um, so we think of most non-living things as not being able to evolve, right? A rock or a desk or your shoe, they cannot evolve. So we kind of associate something that's evolving as being alive. Um, and they possess nucleic acids. Viruses have nucleic acids, so they encode genetic information, and that's how they evolve. Um, so they have certain qualities or characteristics that we see in living things that we don't see in non-living things. So it's a little bit confusing when we talk about viruses as to whether they are living or non-living, because they do share some characteristics with living things, but there's also some characteristics that they possess that are not something that we find in living organisms. So many agree that it's kind of preferable to describe viruses as infectious agents instead of referring to them as organisms, because organisms refers to kind of something that's alive. So we refer to them as infectious agents and kind of resist referring to them as being alive or dead, and therefore we should describe them as being either active or inactive instead. So infectious agents that can be active or inactive. Now, how are viruses classified and named? So there's what's called the International Committee on Taxonomy of Viruses. So there's a whole committee that works on developing the criteria and the naming conventions for viruses. And that's to help to limit confusion. Because before this committee ever came around, it was kind of a little bit haphazard and often misleading the way the viruses were named. So, for example, hepatitis A, B, and C. So you have hepatitis A, hepatitis B, hepatitis C. There's 3 different viruses, but they're not related to each other, but they're all called hepatitis A or hepatitis B or hepatitis C, right? So the A virus and the B and the C viruses are not related to each other. What they have in common is that they all infect and kind of damage the liver. So they were given those names because of the organ that they tend to infect. But it can be kind of confusing as far as how to treat it because they're very different viruses. They have very different properties. So now the viruses tend to be grouped according to the type of nucleic acid that they have. So nucleic acids, once again, are your DNA or your RNA molecules that store the genetic information. They can also be classified by what's called the capsid assembly, and we'll talk about the capsid in just a moment, but this is kind of a protein coat, and it can have a specific structural arrangement to it. They can be classified according to the presence or the absence of the envelope. So the envelope refers to a membrane that can be around the virus. Now, it's not a cell, once again. It's not something that the virus is producing on their own, and we'll get to that too. And we can also talk about the genome architecture. So whether it's DNA or RNA, but also whether it's single-stranded or whether it's double-stranded. So you can see here in this diagram that they're talking about DNA viruses, and the first thing it's asking is it double-stranded or single-stranded? And then it goes down, and is it have an envelope or it does it not have an envelope, right? And then it'll give you different groupings for some of these organisms. And so these are your DNA viruses, here's your RNA viruses down here. So this is kind of a little bit about how we classify and name viruses. All right, so viruses can come in a wide range of sizes. So in this image here, you're seeing in this kind of teal color a yeast cell. And then we have bacterial cells in pink and kind of purple here. And then we finally get to one of our larger viruses here, the panda virus, which is just about as big as 1 nanometer as our streptococcus, which is about 1 nanometer as well. So the largest of the viruses can be as big as a bacterial cell. But then you start going down and down and down, and you get to your influenza virus, adenovirus, and different bacteriophages, which are viruses that infect bacteria, and our yellow fever virus. And you can see how tiny relative to our initial largest virus, so how small they can get. All right, so wide range of sizes across the board for these viruses. Now, I mentioned that there is the capsid, and this was one way in which we can kind of classify or name viruses. So the capsid is a protein shell that packages and basically protects the genome and also accounts for kind of the bulk of the mass of the virus. And the shape of the capsid is based on how the individual kind of 3D subunits, which are called capsomeres, are arranged. So the capsomeres build the capsid, and they may be made from a single type of protein, or it could be a collection of different types of proteins. But unlike most cellular proteins, viral capsids often exhibit what's called self-assembly, meaning the amino acid sequence determines how the final proteins kind of fold and come together to make the larger capsid structure. So self-assembly is basically that no energy is needed for the capsid to assemble. And how this kind of works out is there's still a little bit of mystery around it, but the mechanism is still not completely known. But it has to do with the type of proteins, the amino acids, and how they come together is that self-assembly. Now, in more complex viruses, some of them actually use the host cell machinery to put the capsids together properly if they're much more complex. Now, because the capsid is such a central part of the virus and how it forms, you can kind of tuck this in the back of your brain that this kind of represents a potential target for antiviral drugs, right? So if you want to work against a virus and make it inactive, you might want to think about targeting the capsid or that protein coat. So here are some different types of capsids, just to kind of give you an idea of what these viruses look like. So capsids of most animal viruses have either a helical, as you see kind of in the lower right-hand corner here. So the capsid is kind of that green, so it's representing the proteins, and they're showing the capsomere or the protein subunit and how all of those little capsomeres fit together to make the overall larger structure of the capsid. And we said the capsid surrounds and protects the genome. In this case, it's an RNA genome. So this is a helical one, right? So it's kind of spiraling around. You can also have an what we call an icosahedral shape. So these have typically about 20 size, and they look kind of like three-dimensional polygons. But sometimes they can appear pretty spherical, just kind of like a soccer ball looks very spherical, but it's made up of multiple hexagons and pentagon shapes that are put together. And then we have viruses with kind of the less conventional capsids. They're kind of lumped into a group that are called complex. And bacteriophages, which we have here, exhibit a complex structure. So they can have kind of this icosahedral symmetry, but then they have this kind of hypodermic syringe almost on legs that makes up the rest of the structure. And that helps to inject their genome into a host cell. So to me, these look like the old lunar landing modules, like the old things that we used to send on to the moon and would land on the moon. All right, so these are just some basic shapes that we see. Now, some animal viruses have a lipid-based envelope that surrounds the capsid. So enveloped viruses basically are developed when they bud from the host cell, and when they bud from the host cell, so the host that they have infected, and they've reproduced and they've made new viruses, and when those new viruses emerge from the host cell, they take a portion of the cell membrane with them as a coating over their capsid. So the envelope is really from their host cell. It's not something that the virus produces itself. And there are viruses that are what we call naked, meaning they lack an envelope and they do not bud out from their host cell and do not take the membrane of their host with them.